Hi everyone, and welcome to the 9th Field Notes Live event. My name is Zoe Ash, and I'm a soil scientist in Wasilla, Alaska, and the Field Notes Alaska Region representative. As a regional representative, I serve on the Field Notes Review Committee. The Review Committee solicits and selects topics for each webinar. We have selected 2 exciting topics for you today, and we encourage you to ask questions at any time using the Q&A panel. The Q&A panel should open by default. However, if for some reason your Q&A panel is not open, simply click on the question mark icon located on the right side of your screen. For closed captions, turn on the live caption button located in the lower right corner. Today's session is being recorded. Recorded sessions are available in Teams on the Field Notes channel and the National Soil Survey Center's YouTube channel. Again, thank you for joining us. I hope you enjoyed today's session. With that, I'd like to turn it over to Dave Hoover to tell you a little more about today's webinar. Dave.
Thank you, Zoe, and welcome to all of you that are attending. Hope we're getting more and more people each time. I was looking forward to this session today because for most of the morning I've been talking about the things that seem to dominate our lives these days, and that's talks about COVID and talks about reopening government and accommodations and telework and all those other types of issues that we seem to talk about a lot more than science. And I thought, I need to come to a science webinar to kind of reinvigorate my mind and put it back on that science track. So that's why I'm so glad that we have these webinars, so glad that we're on our 9th one, and that we continue to be able to present some great things that are going on out in the field and highlighting the science that we do. So without any more excited talk, I will turn it over to our first presenters today, Lizzie Carp and Erin Kreutz from the state of Washington, my birthplace, and they're going to be talking about challenges and successes mapping the Olympic National Park. Turn it over to you, Lizzie and Erin.
All right, thank you, Dave. All right, is that showing up?
It is, just put it in presentation mode.
Okay. Hi, everybody. My name is Lizzie Carp. I'm a soil scientist in Olympia, Washington.
And I'm Erin Kreutz. I'm an ecologist based in Mount Vernon, Washington.
And today we're going to be talking to you about the 2021 field season of our initial soil survey of Olympic National Park. We've been given 5 years to map 920,000 acres. We're doing it the traditional way as opposed to a digital soil survey, and we're doing it at Order 3. And in this presentation, we're going to be talking to you about differences in climate that we encountered this past season that motivated changes in the original concepts that we came up with. And we're also going to be talking about challenges and successes that we've had this year. First, I will introduce you to the crew. So in the upper left is Eric Dahlke. He's the office leader here in Olympia. To his right is Erin. I'm to Erin's right. And down in the lower left is Max. He is the project leader here in Olympia. And to his right is Zach Allen, who is an archaeologist with the National Park, and he's been helping us with compliance. And this year we were lucky enough to have 2 awesome drop-ins from Oregon. In the upper right, up in the tree, is Andy Neary. He is an ecologist out of Redmond. And in the lower right is Anthony Calora, and he's an office leader based in Salem. So one important thing to understand about this park is how variable it is in terms of climate, topography, ecosystems, and parent materials. If you look at that map on the right, there is a black line that's about 50 miles long. And over that distance, you can go from sea level to about 8,000 feet in elevation. You'll see landscapes from ocean beaches to alpine glaciers. You'll see ecosystems from temperate rainforests to alpine meadows. And you'll cross over 4 different soil temperature and moisture regimes. And you'll notice that that's a little bit different from what you see on the map. It's changed since then because we got rid of the per Udic moisture regime. We didn't have soil moisture sensors in those areas, so we just didn't have the data to support it. We lumped that in with Udic. And we also ended up lumping xeric in with Udic for reasons that we're going to talk about a lot later in this presentation. But just know for now that there's a huge range in precipitation in this park, going from about 250 inches at the top of Mount Olympus to just 20 inches in the rain shadow. These differences were really noticeable going from our first season to our second. So in season 1 in 2020, we started out in the Quinault region of the park. And if you look at that little map on the lower left, that is where the black rectangle is. So it's on the west side of the park, meaning that it receives this maritime influence, making it really, really wet. And so we were working in the rainforest, and we were seeing climates ranging from isomesic Udic to cryic Udic. And then in 2021, we went to the opposite side of the park. So we were in the rain shadow. And those areas in the map on the lower right part of the slide that are shaded in orange and pink, those are the driest parts of the park. And you'll see that they're still called xeric on the map, and we have since changed that to a dry phase of Udic, which again, I'm going to explain soon. But they were still very dry, and we spent a lot of our time there this season. And those climate differences played a really important role in mapping decisions and correlation decisions that we made this season. So what is the deal with this Udic dry phase? We were expecting to find xeric soils in the rain shadow based on two things. We had Forest Service mapping on the adjoining survey that said that the soils were xeric, and we also had a climate model that we inherited from a previous employee that said the same thing. But once we got out there, we found a mixture of observations that were Udic and xeric, as you can see on that air photo on the lower right. Another source of ambiguity was that we spent a lot of time on these mountaintops that were really dry and sparsely vegetated. And we originally thought that we were going to call those xeric, but as we spent more time up there, we were thinking about whether they would actually meet the xeric definition because they're covered in snow for a lot of the year. And Erin is going to talk more about that.
As we've been teasing our development of the soils legend, we've been discussing how this is going to work into our ecological site development within the rain shadowed areas. And because we have these really dry conditions, it's been really hard for our team to sort out how to handle this question. So we've really had to resort back to the definition of what is xeric. And to meet that, the soil moisture control section needs to be dry for 45 or more days following the summer solstice. And being able to meet that criteria has been difficult for us to achieve for most of these areas. And while it's probable that we may not receive precipitation for 45 or more days in the growing season, it's less likely that these areas in the soil moisture control section are going to be dry. We are often fed by snowmelt throughout the growing season that are giving moisture to these vegetation areas, and especially within our high elevations, their growing season might be less than 45 days total. And this has really helped us kind of justify moving to this Udic dry. So we're really resorting on creating a lot of phase breaks into developing our ecological sites to help account for these huge variations in moisture. So we're going to have phases that range from dry to typical to moist. So one example, the photo on the upper right is an example of our typical Udic forest. And it's probably the forest that we usually picture when we think about Olympic. It's a very lush forest, very high productive trees growing there, lush understory with high diversity and a closed canopy. The photo on the bottom right is probably something we don't picture when we think about Olympic. It's a very dry, low productive forest, scrubby, sparse understory, and we even have madrone growing in these sites. So it gives us even more versatility for creating these ranges within the Udic moisture regime. It's also helping us to create consistency between what the park products on vegetation classifications are and how we're going to deliver our ecological site product. The park developed a very robust vegetation classification in which they're also using these range of phases and moisture modifiers within their discussions. So we're hoping to create a really good crosswalk between our products and what the park has already produced, so that it's more user-friendly and more valuable to our end users. And we're working on really refining what our indicator species are to help create these moisture breaks and define where they occur on the landscape.
One example of how we're going to use these phases is in our mountaintop map units. So the black square shows an area that's eutec, and the red square shows one in the rain shadow that is eutec dry. So on the left, that is a photo from the eutec area. You can see that the image is split into two halves. So just look at the air photo on the right for this moment, and you can see that it looks pretty eroded. There's a lot of scree and exposed bedrock, and the ridges are pretty narrow. And in the left half of that image, there is a raster, and that is from a predictive vegetation map that the park gave us, which has been really helpful. And the grays and the white show unvegetated surface, and you can see that that is dominant. And this is a pretty typical eutec mountaintop. On the right hand, there is an image of a eutec dry mountaintop. And you'll notice in the air photo that it looks less eroded, the ridge is broader, the tone is different, it's a little bit browner, and in the vegetation raster, you'll see that there's more color, which is indicating actual vegetation, most of it different types of meadows. So you can see the differences there.
Our ecological site development for meadows is even more complicated, and we're going to need to include even more phases within the eutec moisture regime. We have such a high variability across our meadows in the park, and they range from very wet marsh marigold and sedge dominated meadows to extremely dry juniper and fescue dominated meadows. And we have a bundle of meadows that are in between these. So it's necessary for us to add even more phasing criteria for these meadows. So we're looking at all soil properties to determine where these phase breaks should occur and what abiotic factors are really influencing these meadow sites.
So in addition to the technical challenges of figuring out our climate questions, we also had some challenges that we faced in fieldwork. The Olympics can have kind of gnarly weather at times, especially in the spring. It can just be really wet. Sometimes it's smoky. The terrain is beautiful, but it does make for difficult hiking sometimes. And while there isn't any seriously dangerous wildlife in the park, it does have particularly aggressive deer. Salt is a really limiting nutrient for deer in the Olympics, and it happens that the human body is a great source of it. And you can imagine what kind of hijinks this leads to. In this picture on the right, there's a deer eating one of Max's shirts, and that was just one of 2 shirts it ate that day, as well as an entire Ziploc baggie full of pesto. But in spite of all this stuff, we had a really good time. It was beautiful out there, and I think we all felt pretty lucky to be out there. So here's the progress we've made thus far. Despite COVID, we've had 2 productive field seasons. We have completed 20 trips in total, 9 in the backcountry and 11 in the front country. We've documented 300 sites and pet-ons. We have developed 75 components and ESDs and 40 map unit concepts. We've completed mapping for 270,000 acres so far. And at this point, we have a pretty solid understanding of soil-landscape relationships in the southern and northeastern portions of the park. This coming field season, we're going to be focusing on the northwest portion of the park. Hopefully, work is going to be less impacted by COVID, but we will see. We're going to be planning longer trips going deeper into the backcountry, because most of the shorter trips have been taken at this point. Luckily, we have some llamas who are going to help us pack this year, hopefully. And we're going back to the rainforest, but it's going to be a different part of the park. So we're expecting to find a new set of climate, soils, and vegetation. And we look forward to sharing what we find with you next year. Thank you.
Thank you both. Great presentation, and we do look forward to continuing these and having another presentation by you both again. We do have one question that came in, and that is your veg raster, is that Landfire BPS, or is it something that the park developed?
The park developed it. And if you want it, we can send it to you, I think.
Yeah, it's still a draft copy, but I think they're finalizing their publication of it soon. But they developed a pretty intense vegetation classification for the park over the last 10 years with, I believe, over 1,000 observations they collected.
And did you have time or opportunity to put in any data loggers for soil moisture or temperature, or are most of your moisture regime analyses done based on current vegetation?
A lot of it has been done with current vegetation. We did have sensors around the perimeter of the park, and I guess maybe that was because of access issues. Erin, were you there at the time those were placed?
Not when they were deciding where they were going to be located, but we did have moisture sensors out for a few years in the beginning when we first started working out there in 2015. But we had some failures with keeping the sensors active for an entire field season and some failures out there. So while we have some really good data from those sensors, we've also weren't able to have a full, complete picture of what's happening out there with those.
Okay, and you obviously got somebody's interest because somebody asked, are there any anticipated detail opportunities?
Ooh. Am I allowed to say? I don't know if it's finalized.
Of course.
I know that we're looking for an ecologist detail. I'm not sure about soil scientists. Do you know, Erin?
I don't know officially, but please, if you're interested, give us an email or please contact our office leader, Eric Dulke, and he can help you with that.
Great response. And we'll got one more question. For clarification regarding the use of phases, are you phasing the soils so that similar soils can be correlated to different ecological sites, keeping one soil in one ecosite? Or are you phasing the ecological sites with one soil possibly correlated to 2 or more ecological sites?
It's a little bit of both. For our soils that we originally tagged to xeric, we're now creating a dry phase for them. And then mostly you're going to see a lot of these phases described within the ecological site.
Okay. And I'm not seeing any more questions come in, so thank you again for your presentation and your responses to all the questions. If more questions come in, we can certainly have those addressed and we'll get them posted.
Thank you.
Thank you.
I want to remind folks that the next webinar for Field Notes is scheduled for February 8th. Work with your regional representatives for Field Notes and get your ideas in. If you saw something today that you said, I could report on something similar that I did where I work, or if you saw this and you thought, I think I should report on something totally different than this, please put together a presentation and share through this medium. Thank you. Our next presentation today will be by Geraldine Vega and Milton Vega out of the Tolland, Connecticut office, talking about urban soils of New York. Milton, I'll turn it over to you.
Thank you, Dave. And it will be just me presenting today. I'm excusing Geraldine. I'm going to cover what she was going to do.
I thought we'd still give her the credit, though.
Yep. Okay.
Just a little bit.
She can have some. It's fine. We worked very, very hard to do this, both of us, so it's all good. There you go. So hi, my name is Milton Vega, and I'm a soil scientist out of the Tolland Soil Survey Office in the Northeast region, sorry. And we're based out of Connecticut. And today I'll be talking to you about urban soils of New York. Go. So I'll start by defining what are considered urban soils. So these are soils in areas with high population density, and they have a setting in mostly built environments. These soils consist of mostly human transported materials, human altered materials, and minimally altered or intact native soils. And the first two are known as HAHT soils. Some of the factors that may influence these soils' characteristics and behaviors are the land use history and disturbance, the geography and geology of the area, the extent of impervious surfaces, and the nature of human transport materials or fill materials. And even those all urban soils are similar in global scale, they all have their own unique soil patterns. So the urban soils I'll show you today were described and sampled by NRCS as part of a project in cooperation with Brooklyn College, Rutgers University, University of California, Riverside. And the purpose of the project is to develop soil survey methods for urban areas. And this project was conducted in 11 parks and open spaces within New York City. And the projects were targeted 14 soil series. The fieldwork includes description of soil profiles, the sample collection for soil property analyses, and the collection of a soil monolith for educational purposes. And here's a map of New York City showing the locations selected for the project. So the project was done in two different MLRAs, MLRA 144A and 149B. 144A is within the blue outline, 149B is within the red outline. So 144A is the New England and Eastern New York uplands, and this area has been glaciated and consists of tiltplains and drumlins. And 149B is the Long Island Cape Cod coastal lowland, and this area consists of unconsolidated glacial outwash deposits of sand and gravel. So just so you know, this side over here is New Jersey, so it's not part of New York. And the New York City is composed of 5 boroughs, and we covered all of them. So the 4 bottom sites right here, this is Staten Island. These 2 sites are in Brooklyn. These 2 sites here are in Queens. These 2 sites here are the Bronx, and this single site here is Manhattan. Manhattan's all the way here and all the way up here. Just so you get situated, this area here is where JFK, the JFK airport is. So we start the project with 4 locations, and there are in the blue boxes. Start off with Greenwood Cemetery, then Van Cortlandt Park, Inwood Hill Park, and the Soundview Park in the Bronx. And this project was completed this past fall. So our first site was Greenwood Cemetery. I'll give you a really short history of it. It was founded in 1938. It's one of America's first rural cemeteries and has 478 acres. And believe it or not, it's a really pretty place to just walk around in, and all the locals do so. It's the most quiet place you'll find in all the city. So we hit working out of Greenwood. We did 2 simultaneous projects. So we did soil survey methods for urban areas research project, which is the agreement we have with the universities, and we had a DSP project with different turf grass managements. including fine turf, minimally managed turf, turf under forest canopy, and unmanaged turf. As I said before, we started with the Greenwood Cemetery, so where we targeted the Greenbelt soil series. The paramaterial is human altered and transported materials. It's backfill material. So for those of you that aren't familiar with the horizon naming conventions at urban soils, when you see a carrot, which you can see right here, right next to the master horizon. So when you see that, that indicates that the material has been transported by humans. And in this profile at 63 centimeters, right around here, we can see a dark matrix color and this indicates us it was a previous surface horizon, and it's accumulation of organic matter. And within that same horizon, the structure there is massive. So that massive structure tells me that there's been machinery moving around. So that's disturbance. It's kind of compacted, and you can see some of that relaxomorphic features kicking in. And if we look at taxonomy, I really want you to focus in two main words, the words spolic and anthroportic. So the spolic terms means that the profile has 50 centimeters or more of human transported materials, and the anthroportic subgroup indicates that the soil formed in 50 centimeters of human transported material. And here we have a hillshade where you can see the cemetery lies within a terminal moraine and a glacial outwash plain. So the terminal moraine is the more complex landscape, and the simpler landscape is the outwash. So you remember the photo at the beginning of the presentation? So it was taken from the north side of the moraine. It's a pretty good view. And so in that photo, you could have seen the One World Trade Center. And here you can see the hillshade showing the Harbor Hill moraine. So it goes all the way from Brooklyn, snakes its way up through Long Island, all the way to Rhode Island. So this terminal moraine was the shore of Glacial Lake, Connecticut. Now going from Brooklyn to Van Cortlandt Park in the Bronx. And so in this park, we targeted the Wasilla soil series. And this has a coal combustion ash paramaterial. So in this profile, you can observe that we're using the horizon naming convention with the lowercase u, the suffix u. And what that means we have indicates us that we have presence of artifacts, which you can observe right here in the fragment column. So to give you an idea of the average of fragments for this profile, there were 48%. So we include the artifacts to calculate this. So it makes it sandy skeletal particle size class. And looking at taxonomy, you'll see that since we use the combustic mineralogy class, so that means that's right here. What that means is we had a horizon of 7.5 centimeters or more thick with 35% or more coal combustion byproducts. And we had our weighted average of coal slags was 36%. And here we have an example of the artifacts that we sieved out. So we use sieves, and I highly recommend using gloves as well, because you never know what you're going to find there. And so we found glass, coal slag, ceramic, metal, clay pots, coal. And here we have a hillshade of Van Cortlandt landforms. So this area that was selected, it was sampled within an old wetland. That was right here, right next to an old railroad. And here you can see the bedrock controlled uplands. And this side here also is bedrock controlled, but it has buildings on it. That's why it looks so different. And going from the Bronx to Inwood Hill Park in Manhattan, I'll be talking to you about the Charlton soil series. So here we have a Neolean phase of Charlton. So when you look at the fragments here from 0 to 105, you can see that there's no fragments. So that indicates us we have a possible yielding deposition. And at 113, right here, we have a restrictive feature. And that restrictive feature here is called latchment till. That latchment till is slowing down water infiltration. and you can see that by the pickup in relaxomorphic features. And pay mind to that red parent material right here. That's the lodgement till. So that red parent material is lodgement till that was deposited by glacial activity followed by yielding deposits on top. So yielding deposits is definitely not a glacial process, but occurs after deglaciation. After the glacier has scoured the surface and leaves its soil bare, so it's a prime environment for that soil yielding deposition. And here we have the hillshade for Inwood Hill Park, and you can see the bedrock controlled uplands. And remember that red parent material? So that red parent material definitely came out from elsewhere, and that's from the New Jersey side of the Hudson River. So this is New Jersey, and this is the Hudson River. And our last park that we sampled in this fall season was the Soundview Park in the Bronx. So in this park, we targeted the LaGuardia series, which consists of very deep well-drained soils. And they're basically construction debris intermingled with human transported materials. And so this pattern ended up being a Taxadjunct of LaGuardia, because if it were the soil series, it would have 35% or more artifacts. And this came back being 26% weighted average of fragments. Had it been that 35, it'd be loamy skeletal particle size class. And the weighted average for the artifacts came back being 13%. Had it been 35%, it would have been artifactic, and it would have made the sorry, it would have made the soil series. And this photo, you can see the artifacts described in the profile. And I can't stress it enough. Always use gloves when managing these type of materials. And here we have the hillshade of Soundview Park. It's pretty flat, but you know we know we have filled marshlands, and we can see on the left and right banks of the Bronx River that this is the Bronx River. But this area here and this area here, we can see that's a signature of a shelf of marshland. And since we're discussing urban soils in New York City, I want to bring to your attention that there is an urban soils focus team. And if you have any more questions, you can reach out to Randy Riddle and Greg Taylor. And before we part ways today, take a final look at the soil profiles we discussed: the Greenbelt, the Wasilla, Tolland, and LaGuardia. Thank you very much for your time. Take it away, Dave.
Thank you, Milton. Great presentation. And it's good to learn not only about new soils, but new words that describe soils too. I know not all of us use those types of descriptors, as spolic and combustic, and other descriptive terms. So it's great to hear those and see those, just what that looks like. We do have a couple of questions. First question is, how long do these urban soils take to form? Any idea of the timeline?
I really don't have an idea, but I went to look at the Big Apple soil series, and that even had plakic horizon, and it's very impressive. So I know there's petagenesis working around there, but I really don't know how long it takes. In dredge soils, it can be fairly quick.
Okay, the horizon that you said was coal slag, and I'm not familiar with what coal slag really is. Does that mean that there was a coal plant someplace in the near vicinity that materials were spread out from, or is this where does it come from?
What we think, so remember that railroad, there was a railroad right next to that place. So that came from it's a byproduct of the railroad. So they chucked out the coal byproducts of the burning for the railroad, and they dispersed them there and filled up that wetland.
Got it. Another question, was there an archaeologist on the project with you to help identify or handle artifact discovery at your sites?
There was no archaeologist. Usually we have an archaeologist look at it before when we're working with Park Services, National Park Service, but not for this. And every time we found something, it was just dumped back in. We didn't keep anything.
Okay, safe practice. Another question on the age of the soils is, how soon are you seeing structure development? How old are these soils? And you might refer to, I didn't look long enough at the slide that you had with the profile descriptions to see what kind of structure terminology you used.
So on for example, this Greenbelt, I can't remember right up there. I can go back. Can I go back to my slide?
Sure.
So we can see we had structure from up here. So, and this movement, I don't know how far along it was, but it does show structure.
So subangular blocky is about as much structure as those you were seeing.
Yeah, that's as much as we see.
Okay. Another question. How did you ensure safety of employees when sampling beyond the use of gloves?
Gloves and sieves. And when we were sampling out there, we weren't using masks because it was when we were supposed to be all vaxxed and all. We were okay, but beyond that, we don't do anything else.
Okay, and one more. Starts off with a compliment. Great presentation, Milton. Out of curiosity, did you notice any difference in vegetation between sites built on moved or human-influenced materials versus more natural? All these artifacts, I wonder if they would affect or harm plant life.
Well, I really didn't see any difference between the soils that were natural. For example, the one in Van Cortlandt, it looked pretty uniform between both areas. Between the natural area and the disturbed area.
Okay. I'm not seeing any more questions coming in. So again, thank you, Milton, for your presentation and for your attention to all of the questions. And thanks to all the presenters today. Another great Field Notes webinar. And one last reminder that the next date is going to be February 8th. And I know that an invite will be sent out for that. And we look forward to more presentations then. So have a good rest of your week, folks. Talk to y'all later. Bye.